that is indication, interphase. Okay? You don't see nucleolus once you are in the mitotic phase. Prophase condensation here, prometaphase, that is, arbitrary. Okay, that is not like exact science right there, right? Just because here you see condensation, here you see further condensation. Prometaphase. Prometaphase is further condensed than prophase. Then here you have metaphase, all line up in the middle, metaphase, okay? Then here you have anaphase, separation, anaphase. And then here you have telophase cytokinesis on the opposite end, and then now you see the uncondensation of DNA. Right? Here, the end of telophase, more the cytokinesis right there. Telophase cytokinesis, more like on the cytokinesis part now. Here is like beginning of telophase. Okay. Here you can see loose, uncondensed DNA, but now see like darker, right? What happens between G1 and G2?
Growth.
S phase. What happens in the S phase? DNA what? Replication. So now you have twice the amount of DNA. So here you have, let's say G1, here you have G2. Uncondensed, right? Unpacked DNA, but now replicated. So now you have twice the amount of DNA because it has been replicated, right? So that's why you can see some who are like faded and some who are like really dark. But both are uncondensed, okay? So I have shown you some G1, G2, then prophase, prometaphase, metaphase, anaphase, and some examples of telophase, okay? Early telophase, by the end of telophase, same thing here, now you see the uncondensation of the DNA, okay? Beginning of the cytokinesis. So early telophase, here you have by the end of the telophase and cytokinesis, okay? For the exam, I will have some pictures like this and I will ask you to label them, okay? Identify them. Which one is this? Then you will tell me this is prophase, this is prometaphase, this is metaphase. For the exam, I will not have you deal with the prometaphase, okay? As long as you know the prophase, metaphase, anaphase, telophase, you're good. Any questions?
Yeah, I have a question. So wait, that orange thing at the bottom is early telophase?
This one?
No, the other one, yeah. No,
This one?
yeah, that one.
Yeah.
Early
So-
telophase?
No. Early telophase will be this because they are still somewhat condensed,
Okay.
right? Once they begin to uncondense and division, then that's more like cytokinesis.
Okay.
Yeah. So this one cytokinesis, that one cytokinesis.
Early cytokinesis?
Yes, well, it's really hard to say, right?
Okay.
But more or less like early cytokinesis. Once you have here, for example, early cytokinesis, late cytokinesis.
Okay.
Yeah.
Why would that be late and that's early?
This one you still see some dark in there,
Yeah.
maybe like some condensation still there. It's still working on the uncondensed DNA.
But it looks-
Here, it's just like uncondensed DNA.
But it looks like it's fully separated.
It is separated, but it still see darker in there,
Okay.
condensed, right? Same thing over here. They are like further anaphase, but now you will begin to see the uncondensation.
Okay.
Right? It is not as condensed as here,
Okay.
right? So early telophase, then here you move into the cytokinesis part. Early cytokinesis, late cytokinesis, ready to divide, right? Because now your DNA, it looks like the G2,
Okay.
uncondensed. Okay?
What was the loops?
Like G1, G2. Both are uncondensed, but one it has twice the amount of DNA, it's darker.
And the red were prophase?
Prophase, prometaphase.
The bottom orange one.
Yeah.
The orange. The two round ones.
That's like the late side of it, because, it's like the loose one.
Yeah.
Any other questions? No? Right, be ready for the exam, okay? At least I will ask you for the prophase, metaphase, anaphase, telophase. Okay? At least you will see those in there. At least you will have the prophase, metaphase, anaphase, telophase. At least you will have those. Be ready for the exam. Okay? Promena phase, I will not include it. This is just like I mentioned, it's just arbitrary. There is no like exact science of like, oh, what is the fragmentation of the nuclear envelope? You cannot see that in there. Okay, so here you have this mitosis plant. You have here the prophase condensation. Here you have the prometaphase, more condensation. Here you have the metaphase, anaphase, and the telophase. Okay. The new substrate right there. Okay, some questions, activities together. If cells in the process of dividing are subjected to colchicine, a drug that interferes with the formation of the spindle apparatus, the mitotic spindle, at which stage would mitosis be arrested? At which stage do they remain? When you use a drug that interferes with the formation of the mitotic spindle. Which one? Prophase, yes, prophase. Remember that in prometaphase, the mitotic spindle begins to migrate to the posterior, so it cannot do that. It cannot go to the prometaphase. Referrin A is a compound produced by fungi that interferes with Golgi vesicle transport, Golgi apparatus. What stage of cell division will it inhibit in plants? Why is the Golgi apparatus important in cell division in plants? Golgi apparatus.
The cell wall. So plate, the plate.
Yes, so what phase would that be?
Sure.
Metaphase?
Huh?
Metaphase?
No, further than that.
Metaphase?
Which one?
Telophase?
Telophase, yes. Remember in that end, telophase, then it goes to the whole division, right? In plants, it will be the Golgi apparatus to make the new cell plate. It cannot do that. It cannot form this. In eukaryotic cells, you have the cleavage sphere, microfilaments. In plant cells, you need the Golgi apparatus to make the new cell plate, to make the new cell wall. Okay. You observe a cell that contains a mitotic spindle that is only partially attaching to chromosomes, so one end but not the other end. And discover this is because the nuclear envelope is not properly breaking down, thereby blocking mitosis. In what stage the cell is blocked?
Metaphase?
Which one?
Metaphase?
In order to be at metaphase, it needs to bind properly, right? Here it is not binding because the nuclear envelope is not properly breaking down. There is no nuclear envelope breaking down. So in which stage the cell is blocked?
Interface first.
Where does the nuclear envelope breaking down happens? Prometaphase, yes, the prometaphase. So that's what it is, because it's not breaking down properly, the nuclear envelope. That's what it's supposed to be. And that's what it's supposed to be, the breaking down of the nuclear envelope, where it's not taking place. During which stage are cells likely to check to ensure that their DNA contains no errors?
G2.
Oh, there you go, yes, everyone got it, nice. You read in science about the mutation in fruit flies that blocks the release of cohesin. Remember that, Leroy? Leroy was cohesin? Cohesin proteins on the centromere. So the sister chromatids do not separate. Okay, so there's a mutation in the fruit fly that blocks the release of the cohesin from the centromeres. So the sister chromatids do not separate. So Leroy was not able to take down by, who was it? Was you? No, who was it?
Anaphase can't happen?
Yes, anaphase cannot happen by you. Yeah, by you, right? So she was separated, she cut cohesin, Leroy, and then separated, right? Here, there has a mutation that Leroy refuses to be broken down by separates. There's a mutation, so sister chromatids cannot separate. Cells that contain this mutation will be blocked during which stage? During which stage does this happen? So they cannot move to the, what phase somebody mentioned? Anaphase, they cannot move to the anaphase. So in which stage are they blocked? Metaphase, yes. That is metaphase. They cannot move to the anaphase because this guy is still joining the sister chromatids. They cannot separate, right? So they're stuck on the metaphase. Cytochalasin B blocks the function of actin. What stage of mitosis will inhibit in an animal? What stage of mitosis? What is the actin important? In which stage? No? Yes, there you go. There you go. Cytokinesis, right? Telophase cytokinesis. It will be forming the contractile ring cleavage forward to create the cytokinesis. Division of cytoplasm. Here, it cannot do that because it is blocking the actin. So it cannot create the contractile ring. It cannot make the cleavage forward. Okay, so there will be that telophase. Okay. Nice, really nice. Okay. So expect some questions like that on the exam, right? Make sure you know which phases are these. The sequence, yes, but also the role.
And those questions that are in the exam?
Not the exact questions, but like sequence. Yeah. Maybe one or two. I will throw one or two. One or two of them. Okay. All right. So here you have whole cell cycle. So there are some cytoplasmic signals. This cell cycle appears to be driven by specific chemical signals that are in the cytoplasm, and they drive this process forward. Okay, so we are now on the G1, let's move to the S phase. Now we finish with the S phase, let's move to the G2. Let's go to the mitotic spin. So there are like some chemical signals that they drive this process, and they discovered this by doing these different experiments. They took these cells that were in the S phase and this G1 cell, and they fused them together. And what they saw is that the chemical signals on the S phase, they were then moved as well to the G1 phase, and they drove the process forward to move from the G1 to the S phase. So it drove the cell to move from the G1 to the S phase, go to the next phase. The same thing they did for one cell who was in the mitotic phase, and they fused it with the G1 cell. They fused them together, and now the chemical signals from the M phase, they move now to the G1 phase, and it drove the process from the G1 to the N phase. It skipped the S phase and the G2. It went right into the N phase. So that you can see here that the chromosome, it is unduplicated. It didn't go to the S phase. It went right into the N phase. So there are these chemical signals, these cyclins, cyclin A, cyclin B, cyclin E, and this CDK protein, CDK1, CDK2, that they bind together and then drive this process forward. What stage is next, and so on. So key point from this, in case that you see this question on the exam, it is that the fusion of these cells at different stages of mitosis reveal that they are like key cytoplasmic signals that drive the process forward of mitosis. Stages of mitosis. Okay, make sure you know that. Right, so you have here this cell cycle control, the sequential events of the cell that are directed by this cell cycle control. It is similar to a clock. Okay, and it's regulated by both internal and external controls. Okay, this clock has specific like checkpoints where the cell is going to stop until it receives the signal to go ahead to the next phase. Okay, so here you have these checkpoints. The G1 checkpoint, the cell is in the G1 phase, and that's where you have like the gene expression, right? That's where most cells will be. And then it will go to the G1 checkpoint, and if it receives the signal, then it will go to the next phase, the S phase, replicate DNA. And then it will go to the next phase, G2 phase, and then at that, it will have the G2 checkpoint. On this G2 checkpoint, it will check, okay, there are no errors, okay, you are grown, yes, you have enough organelles for both cells, yes, okay, go to the next phase, the mitotic phase, okay, the mitosis and the cytokinesis. Here you have the end checkpoint to make sure that it's ready to divide. This end checkpoint is what we did last time here, where you have the chromosomes right in the middle on the metaphase plate, kinetochores bound to the macrotubules, the kinetochore macrotubules, and that's the checkpoint. All chromosomes are bound Kinetic course to microtubules, yes. Or in the middle, yes. Okay. Checkpoint, go to the next phase. Separates cause the cohesion, and it goes to anaphase and then divides. So that is the end checkpoint. So for many cells, the G1 checkpoint, that is the most important one because if it receives that signal, more than likely it will go through all the other phases and it will be able to divide, unless there is like some mutations that could prevent that. The G0 phase, those are the cells that they do not receive the go-ahead signal from the G1 checkpoint to go to the whole cell cycle control, but rather they will exit the cycle, the cell cycle, and that will be in a non-divided state, quiescent state, senescent, resting cells. That is the G1, the G0 phase. Unless they are like brought back into it. So some of them are reversible, some others are not. Okay, some are like fully terminally differentiated. Think of, for example, like neurons. Okay, they are like these quiescent states, quiescent cells. Other cells will be in the G0 phase for a long time, and then once they receive those factors, those signals to bring them back into the cell cycle, then they begin to divide again. Because, for example, like liver cells, the vital sites, they can stay for a whole year without dividing. So that is the G1 checkpoint. So the cell receives the go-ahead signal, it will go and divide, more than likely. If the cell does not receive the go-ahead signal, then it will exit the cell cycle, and I will go to the G0 phase, quiescent state. Okay. So stop and go signs, internal and external signals at the checkpoint. So an example of this internal signal is that the kinetic course not attached to the spindle microtubules, then they will send a molecular signal that delays the anaphase. Well, that's what you guys did over here. If there is a chromosome that is still missing, is not attached, not equal tension, and so forth, we'll send a signal, separates, hold on, do not come and cut cohesion. Okay, so internal signals. External signals are like growth factors, different proteins that are released by other cells in order to stimulate the growth of other cells. Okay, so those are the growth factors. Examples of these growth factors is the platelet white growth factor, PTGF, and that stimulate the division of human fibroblasts. So there are like some proteins, there are some growth factors that will stimulate the growth of specific cells. Also an example of external signal, it is density-dependent inhibition. In other words, that if cells are too crowded, then they will stop dividing. Okay, there is no more space. Think of like real estate. There is no more space to divide, so we need to stop dividing. Okay, so density-dependent inhibition. The other one also is anchorage-dependent, in which the cells must be attached to the substratum or the surface in order to attach and then divide. But cancer cells exhibit neither density-dependent inhibition nor anchorage-dependent dependence. So, for example, here, normal cells, they exhibit anchorage dependence. In other words, they need to force bind to the substrate, to the surface, in order to be attached and then grow and divide. Okay? And they also exhibit density-dependent inhibition. In other words, once they reach all the surface, there is no more place to grow, they will stop dividing. Okay? Density-dependent inhibition. But if you remove some of the cells, you clear some space, and now there is more space for them to grow, they will continue to grow and divide. Density-dependent inhibition, right? So they will come and clear it. Cancer cells do not exhibit this. They can grow, divide, without being attached to the surface, and they can grow and divide even though there is like no more space to divide. So there is no density-dependent inhibition, and there is no anchorage dependence. They divide like crazy, yes.
Is density-dependent inhibition like a feedback loop, kind of?
It is, yes, it is like a feedback because it will tell them, right, hey, there is no more space, we're like so crowded, we're like negative cells, there's going to be like competition, so let's stop dividing. So
Like
it sends a signal.
Negative feedback loop?
So negative feedback, right, it will be because it will send the opposite effect, so there is no more space, right, so they will start dividing.
Oh.
Positive feedback is ones that is going to induce them to divide more and more and more. So that will be more like a growth factor.
Okay.
Yeah. So if you clear that, then now there is more space, and that will send a signal to drive to grow again. Okay, so here you have an example of some breast cancer cells. They are just like dividing like crazy. Those are like cancer cells. They will respond to the cell cycle control. They will go through those checkpoints. Think about like those checkpoints If you're in Mexico, right, you need to come over here, you need to cross the border, you ask for your papers, documentation. Do you have any documentations? No? Stay like over there, right? Yes? Come, right? Go to the next phase. Cancer cells, they don't care about that. They don't care about checkpoints, they just run through that. Okay? So they just divide like crazy and crazy. It's like cells going wild. Right? So they don't respond to the checkpoints. They respond to the checkpoints. They will have certain mutations. They don't need these growth factors, or they even make their own growth factors to grow them. They will grow even though there is like no more space. They will just continue to grow until they kill each other, right? Of course, right? Because there will be a limit to the nutrients and so forth, to the space that it can actually hold them, right? Or kill the host if it's like in patients. They will need like that. They will not need that anchorage dependence, so they will just like keep dividing, even they are not bound to the surface. So that's what it is. So these loss of cell cycle control in cancer cells, that's why cancer cells do not respond normally to the body's control mechanism. So they respond abnormally to the cell cycle control. Cancer cells may not need these growth factors to grow in the body because they are making their own growth factors, or they are making their own pathways that activating these pathways that these growth factors supposed to activate. So they're making that without the need of that particular growth factor. And also they may have an abnormal cell cycle control system that it doesn't need at all. So here's basically just a figure showing you about this. For example, here you have a tumor that grows from a single cell. And then you have like these cancer cells that are invading this neighboring tissue. Then cancer cells spread to the lymph and blood vessels, and it goes to the other part of the body. Then cancer cells may survive and establish like a new tumor in another part of the body. Right? Basically just the progression of this cancer. So first it begins to grow, invades the tissues, neighboring tissues. By doing that, it will destroy the function and the architecture of this tissue. And then they will become metastatic. Right? Remember that we mentioned what is metastatic is like beyond just being in one place, and it begins to migrate. So these are like metastatic tumors. They are going to invade other parts of the body, other tissues, and as they do that, they destroy the architecture of that tissue and destroy the function of that tissue. Metastatic tumor. Once you are in there, like stage 4 cancer, there is practically nothing that you can do. It's just like improving the quality of life of that patient. While you are in this phase, obviously the best option will be like surgery, remove that tumor that is benign. It is not like metastatic tumor. Just remove that tumor because it is enclosed by the extracellular matrix. In this case, like for example, fibrosis might be good because it will like protect the host, it will enclose them. Then just remove it, like surgery, right? Will be one of them. In this case, none, right? Here, fibrosis may even play a role because on fibrosis, you also have these cells that become more like mesenchymal. So you have like epithelial to mesenchymal transition, where normal epithelial cells, they begin to lose their phenotype and not anymore anchor with one another, and now become more like migratory cells, more like mesenchymal, and begin to express a new phenotype, and they begin to migrate. That's basically what it's doing, right? So, surgery, once you are like right here, maybe you will have like chemotherapy. It will kill these rapidly dividing cells, but it also will kill your normal rapidly dividing cells. You will lose your hair, you will lose the epithelial cells in your gut, begin to lose your appetite, and so forth. Other options will like radiation therapy, like high energy beams. Other options will like cryoablation, like insert needles or probes and then like gas to freeze this particular tumor. Other options will be like, for example, immunotherapy, using your own immune system to T cells and then do the CAR T cell therapy, the chimeric antigen receptor cells. Basically have these T cells from your own body, modify them so they can express this antigen against a receptor for a particular tumor, and nothing else from this body, just the antigen from that tumor, and then reintroduce those CAR T cell therapies and go and bite them and attack them and kill them. That's
awesome.
Therapy. It will be amazing, right? Having these your own cells, T cells, and then modify. That's like chimeric, like CAR T cell therapy, and then they begin to express antigens just for that tumor. Reintroduce them so they are primed now, and they go ahead and attack that, only that, and nothing else. Immunotherapy, you have, for example, like hormone therapy as well. You have, like, for example, like VEGF treatment, anti-VEGF treatment, because VEGF is important for angiogenesis to make new blood vessels. And these cancers, these tumors, they're basically making their own blood vessels because they are going to steal the other cells from their nutrients, right? Glucose, like, give me the glucose, I need to grow, right? So it's going to starve the other cells. So what they do is think of, for example, like back in the days, in the medieval times, whenever they were like conquering cities, you attack the other kingdom, what do? Starve them to death. Cut all the roads so they can, like, basically, they don't have any way to grab the, like, the nutrients of their stocks, you know? And force them to get out, right? That's what they do. Here, basically, what it's doing, the anti-VEGF treatment is like stopping making new blood vessels, so they don't have no way to grab nutrients and make them stop, right? And you can see, like, the new blood vessels that the tumors make, they are more like spiky compared to like normal, your own blood vessels. So you introduce this hormone therapy, anti-VEGF, cut the formation of new blood vessels, so the tumor will not begin to grow more, and will not starve your other healthy cells around it. So really amazing, right? But still, we are not there yet, right? We are not there yet in the sense that cancer is still like killing people and many deadly cancers as well. So yeah, different options, different treatment options out there. Okay, so last set of questions. During fruit fly development, mitosis occurs without cytokinesis. This will result in what? If you have mitosis without cytokinesis, what is going to happen? Cells that are initially small, cells like a nuclei, cells with more than one nuclei, destruction of the chromosomes, or cells lacking an S phase. What do you think that's going to happen?
Working eyes.
Which one?
Besides C.
C? Who says C? Leroy? Yes, cells with more than one nucleus, right? Why? Because he just went through the whole S phase, replicated the whole DNA, right? And that's the role of mitosis. But then cytokinesis comes, you want to separate into cells. But cytokinesis cannot take place. So now you have the same cell with the duplicated DNA, right? Two nuclei. What's her name? Radia? Okay. Next question is for Radia. Radia, what do you think?
Balkis.
Sorry.
My name is Balkis.
Balky, come on. Why do you think a cell will want to undergo multiple rounds of DNA replication and mitosis without ever dividing? Right? What sort of advantage could this give? You have a cell with multiple nuclei. It will allow the cell to generate more energy to metabolism. It will allow many copies of DNA to increase and hasten genetic expression. It will give the cell more membrane surface area. It will allow cells to grow larger and take in additional nutrients. What's that? That's what it says. It will allow cells to create increased genetic variability. Which one? D? E?
B?
B? E. E as in Edward?
Yeah.
Oh. Well, that says no? No. Okay. All right, so you have more DNA, right? What is going to happen with that DNA? What happens in
More genetic expression?
more genetic expression? Yes.
Oh, cool, that's right.
Yes, you will have more DNA there, so now you can have like twice the amount of proteins that you have there by doing gene expression. Right? So lots of protein expression, that's what means hasten. Increase and hasten genetic expression. Good. Errors in mitosis are rare, but they do occur. In non-disjunction, one daughter cells gets both sister chromatids while the other one gets none. This could be directly caused by what? Which one? E as in Edward?
D.
D, yes, failure macrotubules to attach to the kinetochore, right? That's what we mentioned right here in the example that I showed you. Yeah, the example that we did here the other day and the example that I mentioned just couple of minutes ago. Right.
What is the metaphor kind of,
What was that?
what is that again?
The kinetical?
Yeah.
Okay, so the kinetical. Sister Chromatins, come over here. Come here.
It was Carlos and
Come on.
Steve.
What is Gaspar? Gaspar and what is the other guy?
At the side of the egg.
Carlos. He just left. Oh no, he was here? Okay, yeah, come over here.
So he has two sister chromatins and then you need all those.
Cohesion? Separates?
Oh, wait.
Remember, like, imagine the egg and then
Yeah.
Okay, in the middle, cohesion of the sister chromatids. Okay. So sister chromatids, cohesion, joining the sister chromatids, but on the centromeres of each of the sister chromatids, it is kinetical. Okay, kinetical. That is a protein right there. Okay. So the microtubules will come and bind. Who were the microtubules for you? It was, I think, Didi? That's her? Okay, come. One kinetical. And who was the kinetical for you? The microtubules?
This two.
Yeah, you pointed over there. There's somebody over there.
Wait, I know I had you next to me, and then they were after
They
you.
were the microscope.
They were the microtubule organizer center, right? You were the microtubule organizer center. Okay.
What? Oh, I guess I'm not